I don't know about the masterclass business, but probably the thoughts of the last 18 months and what's been happening with nitrogen and just the questions people have been asking about, why of the results of the last, I suppose, 3 wet seasons have been the way that they are, ending up with last season with a dry finish and low protein. And people have, I think we understand reasonably well why in a very wet year with a wet finish we end up with low protein. But when you come along with a season where we've basically got no significant rainfall across a lot of the wheat belt and the vertisol soils after July, and it's running on subsoil moisture, why did we end up with low protein? And I've done a lot of thinking about it, and this is basically my thoughts. To a degree, it's a data-free zone. It's not based on field research, but just a summation of my thinking. So I'm quite happy to be challenged and things thrown at me if people don't agree. Just one of the first comments that John asked me to speak about is, have we got the foundations of nitrogen nutrition right? And I think, yes, we have. The nitrogen cycle, just like Chris's sulphur cycle, hasn't really changed a lot, other than some people forget to put arrows in some places and some of the feedback loops aren't all that well considered. But I say, in some ways, the soil's telling us in the protein results help. And I'll show you why. And the thing is, are we hearing and heeding what the soil's telling us, or what the crop's telling us that the soil's telling it? And that's really is really the challenge that I put out there today. And I'll show you why. Why was 2012 protein? Well, a poor protein year. Why is it important? That is the first thing I ask. What's the importance of protein? It's really only one crop, wheat. And to a degree barley, we've got a target zone, but as far as wheat goes, why is it important? Why are we worrying about protein? Why aren't we just worrying about dollars per hectare return? We've obviously there's the domestic market in wheat, and this is sort of a representation of a photo that was developed back in the 1980s in an old thing that we're involved in called Operation Quality Wheat that some people might remember, that went on all that time ago. And what was the problem there? The concern over low protein. This is we're just sort of regenerating something that happened in a 20-year cycle by the look of it, but things have improved to a degree, maybe because of droughts, but certainly on average we're still slipping and we've still got this problem. So from my perspective, there's 2 reasons why we worry about low protein. Is one, from the domestic milling point of view, that's what happens when you end up with variable protein to a baking quality. That's Banks wheat, the same amount of dough, the only produced in the same year on the same plots, and the only difference was the protein that was in the wheat. You can imagine an industrial bakery in this day and age, when they buy bags of a certain volume, and the housewife's not really impressed when it's only half full at 9%, and the manufacturer's not too good, not too happy at 14% when it won't fit in the bag. So from a domestic point of view, it's got issues, but also in the long term, our reputation as sellers of quality wheat and being able to compete in those quality markets of 11% plus. So it is important from a wheat point of view, and I'll show you it's also important from a productivity point of view to maintain the protein a little bit higher. So look, I've put together a summary here of the contributing factors that I believe are driving the issues that we're seeing at the moment. And I put number 1 fair and squarely in the middle, that we've lost the elasticity of the organic matter of nitrogen in the soil because of organic matter decline. In other words, the soil can't give anymore when the weather's right. We've got to make the decisions. The soil's not helping us anymore. In the north here, to a degree, we've lived on the capital of the nitrogen that our organic matter can release. And that's been the buffer for protein that buffers against those big yielding years when the soil's given a bit more nitrogen, so it's matched the yield and we've ended up with reasonable quality protein. Why hasn't we seen it before this? Because of the 2000 drought up until 2007. We continued to put nitrogen on out of a bag and built up reserves in the profile. And that buffered us through the first half of the wet part of 2009, 10, 11. 2012, we wore the running out of that residual, and I'll show you that. To go with the loss of this natural organic matter elasticity, we've got a loss. We don't have a high proportion of legumes in rotations to try and replace some of that nitrogen or easily mineralisable nitrogen that may come from legumes. The net the net position after a legume is not necessarily a gain in nitrogen in a lot of cases where you've got high yields, but it is mobilisation of slow release nitrogen that can be released across the season from organic sources. So that gives us a different source of nitrogen rather than just having to put mineral nitrogen in the soil. We've had a recent record of wet summers and high yields. And those wet summers have also given us some other factors such as denitrification on a very significant scale. And I'll show you that. We've had high yields, again, a large extraction of nitrogen, denitrification. Management strategies have not changed in relation to the change in the situation since the 1990s. So there's the challenge, is to what do we do now that things are changing and we're probably in a new era for nitrogen management. And added on top of that, last year was a dry spring, and where the soil moisture was not where the nitrogen was. So we were actually running into the grand growth period from first node period with roots growing in a actual nitrogen deficit soil. Anyway, so if you've ended up with low protein, I'd say you've got one or all of those things have been in play on your farm to some degree. It'd be fairly rare that if you've ended up with low protein that wasn't as a result of one of those factors. So I'll break it down and look at what did the season look like, what is low protein and what's it telling us, how does the protein get into the grain, and I think that's very important to understand, and it's something that in talking to people I didn't really have a really good appreciation of how it gets in there and what are the influential factors. What are the main influences of grade protein and how much does the crop need and when? So what did the season look like? Yeah, it looked like all of that and probably more. Floods, storms, crown rot, you name it, all of it had some sort of influence. But this is a very busy graph, but just concentrate on the colours of red and green. They're the high decile rainfalls. In other words, the high rainfall that was the best rainfalls for the district in 80% of the years. And you can see during summer, over in, better not push the wrong one here, we've got a laser here somewhere. Top button? Right. You can see summer, or the early summer into 2012, lots of red and green. In other words, high rainfall. And very yellow during that period. So we had a full profile, good rain through till July, and then nothing, which means during the period where 80% of the growth of the crop occurs, as far as dry matter goes, it was existing on subsoil moisture. A busy graph again, but this is a typical mineralisation pattern for Moree based on rainfall, temperature, and those sorts of things. This is the estimation of what happened to mineralisation during 2012, and those X's there, I'm indicating where there were potential denitrification events as a result of the excess rain. So during 2012, leading into that crop, we've had a situation where we had a lot of release of nitrogen, but by that February flood event, there was basically nothing left. And I estimate there's probably somewhere between about 40 and 80 kilograms of nitrogen went missing out of the profile that could have been there during that event. So we were short by that much, and look, there's nothing much people could have done about it. We can't delay the mineralisation. That's your organic matter and crop residues from the previous year releasing its nitrogen as it does every year because we had wet summers, hot summers. The problem was it did that, and then we got the flood in February, and all we really had was a little bit of nitrogen maybe coming out later in the year. But At this time of year it's cold, it's not very much of the total, so it wasn't a good situation for nitrogen from that point of view. And I think, you know, I borrowed this from Jim and Jim Laycock's presentation from last week, and I think that reflects in 2012 in the amount of the number of samples that were in the below 30 kilograms per hectare of nitrogen has grown in the full profile, has grown across time as we've gone from, as I said, this period here where we'd build up residues of mineral nitrogen in the subsoil via the drought. We put it in, didn't take it out, and we've had a succession of denitrification events and high yields, and we're running into a situation where we've got a high frequency, you know, somewhere around the 80% of the profiles are now evacuated of nitrogen. The challenge again for this year is we've just about repeated this situation again this year. So we've just got to be careful and aware that that mineralised nitrogen, if you've had areas go underwater significantly, that any nitrogen that accumulated post the start of the fallow, there's a high chance that some of that's either moved deeper or you've lost it. And that just on a local case study, and thanks to B&W Rural, they supplied me some data out from their customers, we can see there's some other factors that are involved here as well. You can see nearly universally low nitrogen in the subsoil, down at 2. I'm not sure we can detect it much lower than that. That's sort of the getting towards the lower limit of what labs can detect. And the types of numbers in the profile, less than 40, which it just shows that last year in this area in northern New South Wales was really no different to the rest of Eastern Australia, as Jim Laycock's information showed, other than there were a couple of situations where we had long fallows and we had nitrogen down the profile that was somewhat protected from some of those denitrification events. Some of the other significances of this data is I am very concerned about the low level of phosphorus, because that's one thing that can affect your nitrogen efficiency when you put it on. So the question is, if it's there, if we're not getting the plant in a healthy situation to take it up, we've got another problem contributing to the availability. And secondly, is some of these high chlorides. If it's deep in the profile and we've got high chloride or high salinity, it affects water uptake, which affects nitrogen uptake. So it can be down there, but it's not available. So if we count that into availability, we're going to underdo the amount of nitrogen the crop sees. And secondly, that's going to land us into a problem with low protein, particularly in lowering the profile. I've put this up quickly just to demonstrate the value of nitrogen down the profile in dry years. This is some of Wayne Strong's data. And look, if you look at these two here, low protein, the amount of deep nitrogen required was massive, and it couldn't fulfill it. It was more than 100% it needed in that year. The two high protein sites are ones that only used part of the deep nitrogen that was available. So it is valuable in those hard finishes to have deep nitrogen there to get good protein. And so if we don't have it there, we're at risk of low protein in those years. From the other perspective, what's low protein telling us? Largely that if we land somewhere less than 11%, we're foregoing yield also. So there's a strong relationship between grain protein excuse me, and wheat and most cereal crops and their yield achievement. So certainly once we get up above 12.5, we're probably optimising yield, nitrogen and water for the season that was available. But if we're ending up under 11.5%, there's very high surety that we've underdone it on yield. And that comes out of this sort of data for northern New South Wales. There's some stuff from Warwick Felton, just plotting relative yield. We normally say an economic optimum's around 90%, and that comes out between 11 and 12% protein. So there is a direct relationship between protein and yield. This is the way it's been classified. This stuff came out of Operation Quality Wheat 20 years ago, and it is just as relevant now as it was then, with a few minor changes that I'll talk about. a few minor, and they're around some of the other issues we're facing with this low protein. If you want to look at it from a risk perspective, if you're under 12.5% in any one season, then Rain Strong's data would say you've got an 80% chance of getting a payable nitrogen response. Ian Holford's data from 92 would tell you that you've got a 91% chance of getting a 10% yield increase and a 66% chance of getting a 20% yield increase. If we're down at 10.5%, Wayne Strong's data would say we were a 100% chance. In the trials he did, there were every one of them respond when you get to more nitrogen when you get down to 10.5%. Ian Holford's data said 92% you'd got at least a 10% by putting more nitrogen on, and 83% for 10.5. So, you know, I always look at things from a risk-return point of view. There's the risk profile. If you're down below 10.5%, then you're sacrificing some yield. And this is the extent you might be sacrificing. If you're at 10.5% and you produced 2.5 tonnes per hectare last year, or say 3 tonnes per hectare, there was possibly 3/4 of a tonne left in the tank in water that was unmatched by nitrogen. So $200 a tonne, you could have spent another probably $75 on nitrogen in that year and possibly done better out of it. But the thing is, I can say that in hindsight, it's a pretty game person to go there at the start of the season. I realise that. I'm not trying to... but it's just an indication of where we're up to. The other thing on the other side is, from a protein perspective, there is a relationship between that and residual soil nitrogen. So once we get above 13%, we know that residual nitrogen is increasing in the soil. And these are a couple of studies, one from Inside Excite at Taluna, where at about 13.5%, you can see the residual nitrogen grew from about 20 to 20 kilograms, which was left in 20 to 40 left in the profile at the end of the season. Once we got above 13.5% protein, it went up to about 140 once we got out to 16%. So anyone that's grown more than 13%, expect there's probably going to be some residual there, and you maybe need to go and quantify what that is. And the same sort of data come out of the stuff from Warwick Felt and Harry Marcellus and those guys in a different study. So these are principles that have been, they've been established a couple of times. But I think the other thing that you look at is if you're down around in the 9 to 11%, you can expect there's probably only 20 kilograms in the profile available. Thanks, Pete. I'll have to go quickly. How and where does it get into the grain? This is a quick diagram that come out, and it's an idealised diagram, but this is basically the path of nitrogen into the grain once we get into that late part of an anthesis, late part of growth and anthesis. You'll notice that the important parts, the big black lines, are the glooms, the flag leaves, the 2 leaves below the flags, and the pathway via the roots that goes back up towards the grain. And I'll talk about that a little bit later. But just describing quickly, grain fill starts about 10 to 15 days after anthesis. Starch deposition starts first and is followed a couple of days by protein. So when the grain it's pollinated, this is the certain sequence of events that takes place. Starches, and this is one of the important things to understand. Starch deposition is derived from photosynthesis that happens after anthesis. It's not pulling starch out of the rest of the crop and putting it into the grain. Whereas protein comes from nitrogen transferred from the accumulated dry matter. So the concentration and the amount of dry matter that's there affects the protein. And they're independent of each other, and starch deposition is more sensitive to drought and temperature during the grain fill period than protein deposition. So they're linked together, but there are some separate mechanisms. And protein deposition peaks before starch, and that's why in a long, cool finish, our protein peaks and our starch fill keeps going, which is diluting protein. So long, cool finishes, naturally we're going to end up with lower proteins. We end up with higher yields, but lower proteins for a given amount of nitrogen that's available in the crop at anthesis. So what environmental influences are there? Nitrogen, nutrition and water balance up until anthesis are important because it's how much is there and how can we mobilise that and get it up into the grain. a high temperature greater than 30 degrees, it appears, is a trigger for, in most crops or most varieties, to turn off starch production. So when that occurs, if it's close up against when the protein fill has taken place, we're going to end up with higher protein. And if it's too early, we end up with pinched grain, because we end up with high protein, no starch, and that's pinched grain. Water does the same thing, and I've got a question mark over Gregory whether 30 degrees is the real number. It seems to fill long after other varieties are trying to hay off. Part of why we end up with higher proteins and higher yields and lower protein out of that type of variety. Protein is largely unaffected by that temperature, and low grain plant tissue N is an indication that we could be having a protein problem. I've looked through that. What can we do about all of this? Season is a large factor. It's up to 25% of what causes grain protein. I look at it and say, and most people look at that and say, it's the biggest factor. We can't do anything about the weather, so how do we manage it? I look at it the other way and say, well, 75% is not season, so as long as we do a good job on that 75%, we've got some way of overcoming what the season throws at us at the end of the year. And that's the reality of what we're dealing with. For any amount of nitrogen that you might have available, 80, 110, 130, we will end up with a range of grain yields and proteins depending on what the weather is like in that anthesis and fill period. If we have a set and forget nitrogen strategy, and I challenge that in the future we probably can't afford to have what we've had in the past is, we'll just put this rate on, it seems about right for the moisture. That's it for the season. Maybe we've got to review that. The other thing I've looked at pretty closely is varieties. This is out of the NBT sites. Which varieties are taking up how much nitrogen? Because one of the things, have we got some genetic variability in the ability to access the nitrogen in the soil and use it? And there appears to be across seasons. I did this a couple of places. So that's the question is what is that ability? One, to take up nitrogen, and two, to distribute it between yield and protein. And I've done a whole heap of these plots to try and sort out, and if someone wants to talk to me, I haven't got time to go into detail, but basically you can see we've got varieties that sit here in the high yield, low protein, and we've got the ones that sit here in the high yield, high protein, and there's a separation between them. There are clearly distinct differences between the varieties that we're growing these days, and that's 2010. You might say, oh yeah, that's 2010, but what about 2011? Those varieties that are common in this area seem to sit in those quadrats quite commonly. We have these varieties that are high yield and a lower protein, and a high yield and a higher protein. And maybe we need different management according to the variety that we're growing. So we can't grow Gregory and Suntop the way we might grow Spitfire or Sunvale or any of those varieties. They need different management, as it would appear. And then I just looked at North Star last season, and what's happened is these ones have moved down. It did not suit the high protein varieties on yield last year, and to some degree your the low protein high yield varieties did better. But one of the things that's common, I've done this plot over 4 years at some sites, and those varieties that sit out here very rarely actually cross that way. So there's something genetic that inherently in the background in the way they set up yield and protein, and if that is the case, then we can manage with some degree of confidence that we can control this. I've done some plots on protein and screenings, and some of them, yeah, you get higher protein, you get higher screenings, you'd expect that. And at a single site, what happens across multiple years, take 2012, just to show this, Spitfire Sunvale and Suntop Gregory, Spitfire Sunvale, they're all they move a little bit this way, and they might move up and down that way, but they do not cross that line, which is the average protein for the site in that year. So they appear irrespective, and you couldn't have got two more contrasting seasons than 2012 and 2010. And as indicated by the proteins. So There is some genetic effect there, and you question what would happen if we went to 100% Suntop Gregory, what would happen to the protein of the region. Would drop significantly just on the varieties, the way they use nitrogen. I've put together a bit of a spreadsheet in trying to say, well, do I go for yield and protein under what pricing scenarios do I trade off yield and protein? Obviously, in selecting a variety, it's not the protein achievement only. There's the disease package, there's the planting window, there's all sorts of things, but that's a choice that people are going to have to make. Do I grow the high yielding, and at what point do I change over between a high yield, low protein and a lower yielding, high protein as far as protein premiums go? I'll just jump through some of this stuff. I'll put in there the Paddock selection variables that I believe are important. Rotation, fallow management, summer rainfall and previous protein. The different pools, I know it's, I'll jump through some of this to some of the bottom line is organic matter. As I said, if we want to grow 3.5 tonnes without nitrogen, we would need, in my estimation, somewhere around the 2.5% organic carbon. Anyone can put up their hand and say they have that in their soil? So if we go backwards down here, if you're down at 1%, then your organic matter is now providing about 40% of what the crop requires. The rest is coming out of your management of legumes, pulses or fertiliser. So there is why the nitrogen rates are important, because if they're 60% of your result, you get them wrong, you're going to end up with lower yield and low protein, or higher yield and lower protein in some seasons. Last year, looking at the results, more nitrogen recovery, better yields and proteins, but there was a problem. What I did here was looked at two different groups of data, 40 to 60 of N versus where they put on 125 at planting. I believe that last year a lot of the fertiliser nitrogen was stranded because you put nitrogen on. We had a wet summer, we put nitrogen on top of a full profile, we get rain on top of it, where can it go? Can't go downwards into the profile. We got into July, we had 80% of the crop growth to go. It was foraging below where the nitrogen was able to get to. So we've got some soils, I'd say, if we haven't had water over them this summer, that have probably got some residual nitrogen from last year's fertilising still there. And it will need to know where it is and how much is there. So, those that had, high nitrogen, this is the percentage of seasonally available fertiliser from nitrogen. So where there was a high percent of nitrogen from soil, in other words, down the profile, we ended up with higher removal. And the more we put on as fertiliser, where we got the 60% came from fertiliser, we ended up with lower removal. That means to me we either lost it or it's still sitting there. And I would say in some cases it's still sitting there. And the ones where we had 125 kilograms of N on, are showing out there that we didn't get high recovery. So recovery wasn't all that good from fertiliser last year. There's a lot of points that we can touch across the years, lots of tools we can use. Generally, unfortunately, we use one, pre-plant soil N, and that's it. There's a lot of other management tools we should consider if we want to really get back and do a good job on protein. I put together one, Rahm's rule of thumb. Everyone will know Ram Dalal. Back in about 2000, he published a paper where at planting you can review, based on your moisture at planting, what your nitrogen rate should be to get various protein outcomes. I've refactored that so you can look at the relationship. So in this case, if I had 100 of available N, and 120 centimetres of moisture, 200 mils of water available, I'm going to end up with 10.5%. But if I've got 200 kilograms of N available for 200 mils, I'll end up at 13%. Just little things that are out there, tools we should consider, rather than once set and forget if we want protein. The other one I question is one that I'm sort of investigating, particularly in relation to those high yield, low protein varieties where we seem to have very little ability to influence them based on pre-plant nitrogen. Can we do it with late foliar nitrogen? It's done in other places in the world to just bump protein up when yield's been fixed. And I've put some data there. It's at 100% efficiency, would only cost $2.50 per percent. So if we were able to put it in per tonne, per percent, it would cost us $2.50. So if the premium was above that, it'd be worth doing. It's not that easy because there's a whole heap of factors. The efficiency of uptake is anything from 25 to 90% based on international research data. But there is some possibility of doing it based on understanding that it is not going to be a lay down every season. There are variables around, obviously if it's taken up through the flag leaf, the upper canopy and through the glooms, they have to be active. So there's a heap of research that could be done in that area to try and understand that. And once we do, it just becomes a simple thing like a calculator to be able to go, well, what yield, what increase do I need, what's the price of N, what's the grain price classification I'm going to jump if I get up, is it worth doing? Based on the risk that's out there. You might be able to put a risk premium in there. So there's ways of working through these. There is potential. 2010 was a great year, and 2011 possibly to actually get this foliar thing to work late. And I'm talking about after flag leaf emergence through to half head full, that late, and it's as a foliar application. There's potential for it to work, but understanding how we can make it work reliably and make you money in at least 8 out of 10 years is the challenge. It is done in other places in the world, so you've got a question that we can't do it every year, but when that year comes along, are we ready and do we have the information to be able to do it? Because it'll make a difference. So thanks, Pete. I know I've run a bit over time, but that's what we don't want to happen. We're the biggest and ugliest and bitiest thing around. We can even get caught up on the simplest things. Thanks, Pete.
Thanks a lot, Chris. I thought that was excellent. I don't know about everyone else. Surely there's some questions. Stu,
Chris, looking at the varieties of your matrices, have you dug into phosphorus in the background as its relationship to varietal phosphorus use efficiency driving yield and then nitrogen uptake? Just having seen phosphorus responses late, where half-time yield equals percent enough protein drop directly related to phosphorus. Just got me thinking, what's the relationship to, is that one of the things? Because sorghum varieties and other varieties, their relationship to phosphorus uptake is can be quite genetically determined.
Look, there's a lot of factors in why those varietal performances are different. That could be part of it. And it's sort of something that I'm sort of taking the next step of trying to understand. And as I understand it, anything that affects the root system, and that might be genetically, it's got a different root system. Certainly phosphorus plays and it has an influence on root structure, and that influences nitrogen uptake. The second part of protein, well, that's the first part of nitrogen, of the protein story, is how much nitrogen that was in the soil ends up in the plant by anthesis. That we know that is affected by phosphorus. That can be genetic, and it can be just phosphorus adequacy to drive a decent size root system with decent activity. The other part is mobilisation of that nitrogen from the dry matter into the grain, which is genetic and it's environmental. It's genetic from the point of view of the enzymes that actually take the proteins out of the grain, break them down to amino acids and have that transferred to the grain, because they've got to become soluble again to move out. So how efficient is that enzyme under the environmental conditions? And to a degree, how efficiently can it use nitrate that's coming from the soil that late in the season, which is another enzyme. So there's a whole heap of factors, but phosphorus, yes, at the base, you will end up with, in some cases, higher yield and lower protein from phosphorus, just because it's limiting yield, and it limits yield. You've got yield before protein, so you end up with a higher protein. So that, ultimately, for those that want to go down that route, growing phosphorus deficient wheat will give you a higher protein, but it won't necessarily put more money in the bank, because you'll suffer yield penalties for doing it. So look, it's one of the unknowns. This whole area is not real well known in this environment, and lots of opinions, but not lots of. It's not an area of rich in facts.
Thanks.
Yeah, Chris, just a comment to Stuart's question there. With the phosphorus placement work that we're going through at the moment, getting the peat nutrition increase into the plant definitely does seem to have that ability of letting the crop recover a lot more nutrient.
Yes.
So it just seems to grease the wheels for the plant. It can recover a lot more, not only peat, but and as a consequence with it. So we haven't done any kind of mechanisms of it, but certainly the recovery of nutrient in terms of the amount of biomass and the amount of nutrient that goes into the plant, it is increased.
Yes.
Any other questions?
Briefly, we sort of use a rule of thumb that says 11.5% protein is probably the maximum yield. But the sun-top Gregory type of thing, it's probably lower than that. Probably
No, it's not. We haven't redone those calibrations with particularly those two varieties, but I would agree with you. There would appear to be optimisation at more like 10.5 to 11.5% rather than 11.5 to 12%, 12.5. So that's one of the things that would be nice to be able to do, is to go back and generate that graph of relative yield versus protein for those varieties that sat up in that top left-hand quadrant, to see if it is really different to the other varieties. Because if it is, it does mean that if you're doing your nitrogen budgets, we're going to base it on possibly 10.5 to 11% rather than 11.5 to 12.5% for those varieties. And that is a big it's a big leap forward. It's been an area of darkness for a while, is understanding what part variety plays. And I don't know whether we really understood it or really looked at it much in the past. I know Hartog was a real issue when it came out, and there's a lot of work went into that. And I suspect this is one out of the same sort of stable. Not that it's the same breeding, but it's coming out of that same area. It's a shock to the system in the way these two the varieties that sit over there in Parla, Suntop, and Gregory appear to handle nitrogen.
Thanks, Chris. So we have one last question on the back.
Yeah, just in regards to urea application, putting on urea, say, a month ago before all this rain, there'd be a higher risk of losing that urea compared to putting it on, say, today and sitting in the profile for the plant. So I would say the plant would be more able to access that urea if you put it on there. What's your idea to that? If
you can tell me what the rain, whether it's going to rain or not, and how much next week, I'll give you the answer. It's one of those things that if you put nitrogen on and it is in a layer of the soil where you have breaking down stubble from last season, and it waterlogs significantly, then there's a significant loss potential. Whether you put it on last week ahead of this lot of rain or next week ahead of probably 25 mils, it's going to give you the same conditions. If the profile's full, then in my estimation, you'd be trying to go a little later to try and avoid summer storms, cyclone residues, and those sort of things, because it does not take long to waterlog when you've got a full profile, compared to if we had a normal summer where we'd have some space for that waterlogging potential to be taken up. So we're at a risky time right now because the profiles are full. Until it starts to cool down and we start to get out of high rainfall events.
No, thank you.
We might just give Chris a round of applause. Thanks a lot, Chris. That was great.